Hey everyone, welcome back to The Explainer. I am so thrilled you're here today because we're going to dive into something really cool, the invisible architecture that literally builds the world around us. We're talking about the molecular building blocks of life. It's kind of like a journey from microscopic elements to these massive natural structures. And honestly, by the end of this, you are going to look at your food and, well, your whole environment entirely differently. Let's get into it.
To kick things off, I want you to think about this seemingly absurd question. Why can you easily boil and eat a soft potato, but you absolutely cannot eat a rigid tree branch? I mean, what if I told you that the fluffy, energy-rich potato and the hard, splintering wood of a tree are actually mostly made of the exact same simple sugar? No way, right? But it's true. The difference between a baked potato and a piece of lumber basically comes down to a tiny, microscopic flip in how their molecules connect.
Okay, let's dive into this. Here's our roadmap for today. First, we'll look at the elements of life. Then, glucose and its isomers. After that, we'll hit the glycosidic bond. Then, building bigger with disaccharides. And finally, polysaccharides, from starch to cellulose.
All right, part one, the elements of life.
So, all organic architecture starts with a really simple foundation of carbon atoms forming strong bonds. But the very first true building block concept we need to lock in right now is the monomer. Think of a monomer as just an individual brick. In biological terms, it's a small, single subunit. Now, on its own, it's just one piece of the puzzle, but its true power, well, that lies in its ability to bond with other repeating subunits.
And when those single monomer bricks snap together, they form what we call a polymer. Try to visualize thousands of individual bricks rapidly locking into place to create this massive solid wall. That is exactly what a polymer is. It's a large macromolecule formed by repeating monomer subunits, all coming together through a chemical process called polymerization.
Now, when we look specifically at carbohydrates, they are formed from just three elements: carbon, hydrogen, and oxygen. But the really neat part is this strict rule they follow. The ratio of hydrogen to oxygen atoms is literally always 2:1. Does that sound familiar? Well, it should, because it's the exact same ratio found in water, H2O, which actually is precisely where the hydrate in carbohydrate comes from.
Moving right along to part two, glucose and its isomers. Let's meet the most famous monomer break of them all, glucose. It's a 6-carbon sugar, but glucose actually hides a pretty fascinating secret. It exists as an isomer. Now, what does that mean? Well, an isomer refers to molecules that have the exact same chemical formula. So for glucose, that's C6H12O6. But they take on a structurally different form. It's kind of a mind-bending concept, right? The ingredients are identical, but the final shape is totally different.
Now, what's really interesting about these ring structures of glucose is this tiny structural variation. I want you to focus specifically on the hydroxyl group, that OH group located right on carbon number 1. Look really closely at the difference here. In alpha glucose, that hydroxyl group is always positioned below the ring, always. But in beta glucose, that exact same hydroxyl group is completely flipped and is always positioned above the ring. I mean, it seems like the most insignificant, tiny structural flip imaginable, right? But as you'll see, in nature, this single variation results in entirely different biological functions. It's absolutely crucial.
Okay, part three, the glycosidic bond.
So how do we actually build with these biological bricks? Well, the bonding process is basically like a highly choreographed, tiny molecular dance. Step 1, 2 single sugars or monosaccharides approach each other. Step 2, their respective hydroxyl groups interact. Step 3, during this interaction, exactly 1 water molecule is removed. And finally, step 4, with the water gone, a super strong covalent oxygen link forms between the 2 sugars. We call this entire process a condensation reaction.
And this brilliantly illustrates the exact mechanism in action. You can just see those 2 glucose molecules coming together, the OH groups interact, and boom! A water molecule, H2O, is ejected right out. And what's left behind is that oxygen atom bridging the gap, perfectly locking the 2 monomers together in a really strong glycosidic bond.
All right, part four, building bigger with disaccharides. So, with the biological mortar now mixed, we see 2 single sugars become a functional pair. To help us kind of consolidate the massive variety of sugars out there, just take a look at this data. We start with monosaccharides, like glucose, which we use for base energy. But when we use that glycosidic bond to join 2 of them together, we get a disaccharide. You're probably going to recognize some names here. When alpha glucose joins up with fructose, it forms sucrose. That is literally the exact same sugar stored in cane sugar that you probably stir into your coffee. And when glucose joins with galactose, you get lactose, which is the sugar found in mammal milk.
Let's move to part 5 and see how this all builds up. Polysaccharides, from starch to cellulose.
Now, here is where we scale up to massive chains made of thousands of units. Remember that tiny alpha and beta isomer flip we talked about earlier? Well, here's where it literally changes the world. When a plant or animal builds a polymer exclusively out of alpha glucose, where that OH group is below the ring, it forms starch or glycogen. These are highly efficient energy storage molecules. Basically, they're the potato. But when a plant builds a polymer exclusively out of beta glucose, where the OH group is flipped above the ring, it forms cellulose. And cellulose does not store energy. Instead, it creates rigid physical structures like cell walls.
So, the crucial point here is understanding exactly why beta-glucose creates such incredible strength. Because that hydroxyl group is flipped up in beta-glucose, forming consecutive 1,4-glycosidic bonds in a straight chain actually requires every second molecule to physically rotate and invert 180 degrees. This constant up-down inversion allows countless strong hydrogen bonds to form between parallel chains. They interlock just like an unbreakable microscopic woven cable. This is what gives cellulose its immense structural strength, and it finally answers our question from the beginning. This is exactly why you can't eat a tree branch. You just don't have the enzymes to break down those inverted, tightly bound beta-glucose chains, unlike the easily accessible alpha-glucose chains in a nice starchy potato.
It is truly incredible how nature uses the exact same atoms, just slightly rearranged, to create both the soft food that fuels us and the rigid timber that houses us. Knowing that simple sugars can be inverted to build towering trees, I'll leave you with this. What other massive natural structures in the world around you are hiding brilliantly simple molecules as their hidden foundation? Keep questioning, keep observing, and thank you so much for joining me on this explainer.